Do you know what technology has recreated this prehistoric beast? It is a technology called cloning of animals. The word cloning sounds very hi-fi, doesn't it? But it is fairly common in nature. Bougainvillea, rose, ginger and many other plants propagated through cuttings are natural clones of a mother plant. Among higher animals, identical twins are examples of natural clones. Identical twins start their lives as a single fertilized cell which divides rapidly. If this cell mass gets separated into two cell masses accidentally, they will grow into two fetuses. This property of embryonic cells to develop into a fully adult animal is called totipotency. But once the animal becomes fully developed, its cells lose this totipotency. They produce only clones of their own kind upon division. For example, a muscle cell will divide and produce clones of muscle cells. A liver cell will divide and produce clones of liver cells. But Ian Wilmut of United Kingdom has produced a technology called somatic cell nuclear transfer to reprogram adult cells to regain their totipotency. Let us discuss what this technology is about. He took the ova of a Scottish black-faced eve and removed their nuclei. Then he took the udder cell of a fin dorset eve. He fused this udder cell with the nucleusless ova to mimic fertilization. These ova with fin dorset nuclei were put inside a foster mother. After 277 such trials, one live birth occurred. Her name was Dolly. Soon after, many such animals were produced world over. Our country also is a proud owner of a buffalo clone by the name Garima. Naturally, now you will be wondering about clones of adult humans. A company called Clone Aid claims that they have produced first human clone by the name Eve. But don't worry, we may not be able to clone a Hitler or Mussolini or Idi Amin because human development involves an interplay of genes and environment. But this technology may bring back many extinct and endangered animals to life. Cloning of dead pet animals also have become a multi-million business. Now you know, cloning of animals involved transfer of nuclei from adult cells to nucleusless ova to behave like a fertilized egg.